Last time we left you with a question: why is testing hERG alone no longer enough? Here's a counterexample. There's a drug called verapamil. It blocks hERG harder than many drugs that turned out to be deadly. Yet it's been used safely for 40 years, taken by hundreds of millions of people, almost never triggering that lethal arrhythmia. Same hERG block, so why the opposite fate? If you looked only at hERG, verapamil would be flagged as high risk and killed on the spot. But in real patients, it has been safe for decades. That tells us something. Testing hERG alone wrongly condemns many perfectly safe drugs. It's a false positive. Verapamil's secret is that it doesn't just block hERG. It also blocks the heart's calcium channel, Cav 1.2. Recall the heartbeat. Repolarization is driven by hERG letting potassium out. Depolarization and the plateau are driven by calcium flowing in. Block hERG, and repolarization drags on, and the cell throws out an abnormal discharge called an early after-depolarization, the very trigger for torsades. But verapamil, by also lowering calcium, holds that trigger down. One pushes, the other pulls, and the net effect is safe. This is called balance block. So here is the truth. What decides whether a drug is lethal is not any single channel, but its net effect across a whole set of cardiac channels. Watch only hERG, and you will both miss truly dangerous drugs and kill safe ones. To see clearly, you must test and integrate multiple channels together. This new paradigm is called CiPA, the Comprehensive In Vitro Proarrhythmia Assay. CiPA rests on 3 pillars. First, in vitro multichannel testing. No longer hERG alone, you profile a panel of key cardiac ion channels. Besides hERG, there is the sodium channel Nav 1.5, especially its late component, the calcium channel Cav 1.2, plus supporting potassium channels IKS, IK1, and ITO. The 3 with the biggest impact on lethal risk are hERG, Cav 1.2, and late Nav 1.5. And the method is still the gold standard from last episode. Patch clamp. A pile of channel data is not enough. The second pillar is in-silico modeling. You feed how much each channel is blocked into a mathematical model of a human ventricular cell, the O'Hara-Ruddy model. Let the computer reconstruct the cell's action potential, and compute a composite score called QNet that directly rates the drug's proarrhythmic risk. The third pillar is human stem cell-derived cardiomyocytes. Using iPSC technology, human cells are induced into beating heart muscle carrying nearly the full set of cardiac channels, validating the first 2 steps in living cells. In vitro, in silico, and living cells. Only when all 3 agree does the conclusion hold. And this is not just science. It saves money. Under the 2022 update to ICH E14 and S7B, if a drug's non-clinical data are solid enough, a clean hERG result under best practices plus a negative in vivo QT, that double negative can replace a costly clinical study, the thorough QT study, or TQT. A single TQT runs into the millions and slows the whole program. Do the multi-channel and CiPA work well, and you may skip it. From testing one channel to profiling a whole panel, adding models, adding stem cells, cardiac safety is becoming a precision engineering discipline. Next time, we go into the most dangerous channel of the set, the late sodium current. Why does its failure put the heart at risk? Follow Channel Observatory, and we will take them one channel at a time.